Welcome to ASM Live at the 115th General Meeting of the American Society for Microbiology here in New Orleans, Louisiana. We are going to talk today about some stories that are important, interesting, and sometimes surprising that relate to microbes and their impact on the world around us. We invite those of you in the audience to ask questions. Just step up to the microphone, tell us your name, and we'd love to have you join in the conversation. If you're listening online, please send any questions through Twitter at #ASMLive. Today, we're going to be talking with James Kramer from Michigan State University. And James works on the microbiome of plants. And an area you've been studying lately has to do with how the microbiome of plants influences the immunity of plants. Could you tell us something about this?
Sure, and thanks for having me on. It's a real pleasure to be here. So yes, I study the plant immune system and how it interacts with all the microorganisms on the plant. And you might be thinking, well, plants have an immune system, but yeah, in fact, they have a very good, robust immune system. And it's based on the premise of recognizing molecules that are self or non-self. So a plant sees a molecule and senses it with these receptors and recognizes it. Is it a plant molecule or is it a bacterial or fungal molecule? And when it recognizes these molecules, it can respond to these molecules. And it's fascinating to me that plants can live in soil, really, because soil is the richest microbial environment known, full of hungry microorganisms. And plants are sugar factories. It's called autotrophs. They can make energy from sunlight and sugar factories, basically. And it's very important for plants to be able to recognize the fact that certain microorganisms are on them to be able to defend themselves so they're not exploited by these microorganisms.
So if they're sitting in this environment with all of these microorganisms, some of which are probably beneficial for the plant, how do plants distinguish pathogen from beneficial microbe?
So there's different tiers of plant immunity, you could say, and sometimes plants will get confused. Plants will recognize a beneficial microbe as a potential pathogen, but different groups of plants will have different sets of receptors, and through evolution, they've evolved these receptors to be able to recognize particular groups. For example, tomato plant and other solanaceous plants are unable to recognize certain pathogens, certain bacteria, based on a molecule called elongation factor 2. And other plants, like Arabidopsis, the plant that I study, the model plant, is able to recognize a particular pattern on this elongation factor 2. And what this does is the Arabidopsis is able to mount an immune response to it. But the tomato plant is not, but it can be transferred. That ability can be transferred over. So it's just through selective pressure in evolution that plants have evolved these pattern recognition receptors to be able to recognize a pathogen from a non-pathogen and mount an immune response.
You know, when we talk about immunity in animals, we talk about innate immunity, this initial immunity where you're recognizing something for the first time maybe, and also acquired immunity where you're responding after you've seen a pathogen before.
Right.
Do plants have that type of memory, the acquired immunity as well?
So plants do not have an adaptive immune system per se. They don't have antibodies like we do or mice do, and they do have the pattern recognition receptors, but they can't be immunized. But however, one thing that we found from our research, which let me add that we're not the first people to study this phenomena of the plant immune system by any means, and people have studied these phenomena before. And what we were able to do, though, is if we remove microbiota from plants and probe their immune system and see how well does the plant respond to a pathogen or a particular pattern from a pathogen, we were able to find that the plants that never experienced seeing a bacteria in its life, for example, are a lot slower and respond less robustly. So you could say that plants are trained throughout their life. Their immune system develops over the course of their life to be able to respond to microorganisms.
So that's very similar to the way we think about the microbiome in animals in the gut in terms of stimulating your ability to respond.
Yes,
yeah. Right? But so how do you have a plant that has never seen microbes?
So that was not an easy task for us to accomplish, actually. We spent many years trying to develop a system where we can actually have microbe-free plants that is analogous to a soil environment, a natural environment. So people have made microbe-free plants before. You can take seeds of certain plants and sterilize them with bleach and ethanol and get rid of microorganisms. But oftentimes people will take these plants and grow them on nutrient agar or in a hydroponic system, say. But what we wanted to do is have something reminiscent of soil that microbe-free plants can grow in, so we can understand when we introduce microorganisms into the system, how does the plant change, and how does the plant, any adaptations that the plant will have to being in an environment with microorganisms, we can infer that this is a function of the microorganisms. So the way that we did this was we take your standard potting mix from a greenhouse, and we sterilize that with a careful heat killing procedure, lots of short intervals of heat killing, and we made a gnotobiotic system, which is a germ-free system, you could say, that has the ability to inoculate, and you can grow plants in the system and analyze with different combinations of microorganisms. How are the plants different?
So you said that there are several pulses of high temperature. Why several pulses?
So
why don't you just autoclave the soil?
So if you just bake the soil for a long period of time, the chemistry can actually change of it. And in some cases, this can actually have toxic effects on the plant. So this has been known since at least the 60s. People have been trying to autoclave soil and grow plants in it and see that their plants don't look so good. So we found that this process of shorter intervals of heat killing will kill all the easy to kill guys on the first pulse, and then some spores and some resilient, more hardy microbes might survive that first heat pulse. But the stress of the heat will cause them to germinate, and then a second pulse and a third pulse will ultimately kill everything. So then you have this sterile substrate, but the chemistry hasn't changed quite so much.
Much like pasteurization of milk.
Exactly.
We have a question from the audience.
Sure.
Hi, my name is Sara Markland. I'm a PhD student in the Microbial Food Safety Laboratory at the University of Delaware. And my question is, my research focuses on whether or not plants can mount an immune response towards human pathogens. I just wanted to know if you had any insight or thoughts on this sort of new field of study that we see coming about.
Sure. So both E. coli and Salmonella, common pathogens that you've probably heard about in the news for food safety reasons, have a tail called a flagellum. And on this tail, they have a particular pattern of acids, amino acids, that the plants can recognize. So indeed, the plants can recognize certain human pathogens if they get on their surface. Yeah, and I think that that's a huge emphasis that we should put on this field of plant microbiome research is food safety. Understanding can plants recognize and how robustly do they mount an immune response to human pathogens? Because say if we were able to have lettuce that can recognize and mount a more robust immune response against Salmonella, E. coli, that could be really important for human health.
You got to be careful here. If you do too good of a job getting rid of Salmonella, it puts me out of work.
We'll work together.
But that is a real interesting thing, because Salmonella and E. coli, you would expect plants might be exposed from fertilizer, but otherwise it's not something that you would normally find out in a pristine soil environment. So we have another question from the audience.
Hi, Good morning. My name is Dr. Lindsey Gilda, and you've been talking about the plant response to these pathogens. Could you just briefly go over what the general response of the plant is once it does recognize a pathogen or one of its beneficial microbiota?
Sure. So when a plant recognizes a pathogen by sensing a particular pattern on it, a number of different things happen. And the plants will reinforce their cell walls. They put this difficult to penetrate sugar called callose on their cell walls. And then they produce hydrogen peroxide in a burst. And this hydrogen peroxide not only is antimicrobial, but it's a signaling molecule, which is perhaps more important and more relevant to the system because it signals to other plant cells like, uh-oh, we've got a pathogen that we need to do something about. And there's also specific genes that get expressed that are defense genes. But a lot of the aspects of immune response and any kind of antimicrobial compounds that a plant might produce, it's still a big topic of research, and a lot remains to be learned. But when a plant recognizes a beneficial microbe, one of the systems that people have studied really thoroughly for this is looking at Rhizobium, which is a type of bacteria that can form a nodule on the roots of certain types of plants called legumes. And it's really interesting that there's quite a bit of overlap in the signaling pathways for an immune response and for a symbiotic interaction. And in order for this bacteria to come in and form this organ in the roots of a plant, it has to actually, stimulation of the immune system is essential for this process. So there's quite a bit of overlap, and it's really fascinating how these different pathways or variations of the pathways have evolved.
You know, when I was learning about plant microbe interactions very early on, people were talking about very specific interactions. Mm-hmm. One pathogen, one plant response. And what you're talking about now is a generalized response,
right,
which is quite different than that.
Indeed, yes. So, like I said before, if it weren't for people and fantastic researchers doing this mono e mono, one pathogen, one host plant system, and understanding what goes on in that interaction, we would have no idea how to interpret this community data. And I think that innovations in sequencing technology, the ability to look at all the genes being expressed and all the DNA in a system in a single reaction is really enabled us to study things in a community context. And I think that we can learn a lot of new things now that we can answer these old questions with new technologies.
We have another question from the audience.
Hi, Dr. Kramer. We know that plants produce secondary metabolites, and many of these are antimicrobial. I guess I have two questions. Number one is, do you know whether these plants produce different amounts of secondary metabolites when they are exposed to more, let's say, bacteria? Number two is, what's your opinion about Chinese herbal medicine in terms of treating infectious diseases?
So I think a lot has to be learned for what sort of secondary metabolites different plants can produce. We're just sort of in that transition now that we're really starting to understand model plants like Arabidopsis, and by understanding how secondary metabolism happens, then we can start to look at other non-model plants like Chinese herbal plants in which you would extract Chinese medicines from, or and I think that plants can produce secondary metabolites associated with defense as part of their immune reaction, but lots needs to be learned, and different microorganisms can stimulate plants to produce different types of secondary metabolites, and in some cases the plant can produce a secondary metabolite, and then the microbe living on it or inside it will take that metabolite and convert it into something else. So it's a really complicated network of interactions, and it's really exciting to be at like the genesis of trying to understand of what these new microorganisms can do with these metabolites.
So in this situation, in the soil around any plant, including your plants that you have isolated in this particular environment, there are lots and lots and lots of different microbes.
Oh, yeah.
Right? So how complex does the mixture of the microbiome surrounding the plant have to be to begin to stimulate these processes?
Well, you can have a single microorganism. You could take just E. coli or Salmonella or a plant pathogen and put it on a plant that has no other microorganisms, and you'll see a very strong response, no doubt. You don't have to have many organisms to stimulate this sort of response. But what we're trying to understand with our research is in nature, that's never happens. You never have a plant that only has one microbe on it. You have a plant that has thousands of different types and millions, tens of millions of different microbial cells on the plant. And so what we're trying to do is reduce the complexity of the environment by taking it into the lab, but maintaining the complexity of the microbial communities. And by maintaining the complexity of the microbial communities, we can begin to understand what's happening from a community context. Does the plant response to 10 or 100 or 1,000 microorganisms differ from how it would respond to just one microorganism? That's one of the main focuses of our research.
We have another question from the audience now.
Steve Lipson. A lot of people, healthcare individuals, talk a lot about proanthocyanidins, epigallocatechin gallate to be used in pills and tablets. These are, of course, plant secondary metabolites. How effective are these chemicals in decreasing the extent of activity of plant pathogens, let alone using these chemicals per se to eliminate human disease?
So studying the importance of different facets of the plant immune response is a huge area of research. And since there's so many different arms of the plant immune response, it's a synergistic effect of many different aspects of the response pathway. So, I think that each secondary metabolite would have to be evaluated individually against a specific pathogen. The answer is they have great promise, but I think things need to be evaluated on an individual basis, and it's really important to do that sort of testing.
So along the lines of that question, James, is your group or are other groups looking actively about the microbiome community and how that stimulates production of some of these secondary metabolites by using a number of metabolomic approaches?
So our group specifically has looked at the production of different hormones that the plant can produce, but we've really focused more on the genes that can be expressed in response to different organisms that you put on the plant. But yes, there certainly are many groups that are studying the metabolites that a plant can produce in response to a microorganism. So our initial focus was developing this system, which we considered like a platform for discovery. I think using this flow pot system, we call it, you could eliminate all the complexity of the environment, inoculate with different strains of microorganisms, use whatever sort of plant that you desire, and then you can study what sort of metabolites are produced on the plant. And there's techniques to try to determine if it comes from the plant or from the microbe, or if the microbe modifies something from the plant, and so on. Yeah.
I think we have another question from the audience, and while we're waiting for this question, I'd like to remind everyone listening online, you can send questions in through Twitter at #ASMLive.
Hi, I'm Nancy Johnson from Northern Arizona University, and it just occurred to me, I'm wondering if you might have some generalized criteria that might be helpful when screening these interactions, because it would take a long, long time to look at every plant microbe interaction.
Right.
Are there certain ecological principles or other environmental principles that might be useful to help us know when to expect a pathogenic versus a mutualistic association?
Mm-hmm. Yeah, I think that it's probably community context specific. So if you have soil from Arizona versus soil from New Orleans versus soil from Michigan, something that might be a pathogen in one environment or have mildly pathogenic effects might not necessarily have a pathogenic effect in another. And it might depend a lot on how much of a particular microorganism is present there. So I think it's really, it's something that has to be evaluated on a case-by-case basis and with focus on like what is the question that you're asking. So if you classify something as like mutualistic or even beneficial to the plant, it's not always beneficial. Something could be beneficial that confers drought tolerance, but the plant's actually a little bit smaller when it has all the water that it needs. So you really need to have, I think, a specific criteria for what do you mean by beneficial or mutualistic, and then simulate those kind of negative characteristics or negative environmental conditions that the plant might experience, and then screen microorganisms in that context.
So along those lines, you can either look from a very broad perspective, the ecological perspective, where you have to deal with these complexities. The other thing is you can take this reductionist approach that ties into using a model plant in a model system and reduce the complexity of the population and see if you can learn some principles from the reduction of that complexity.
Yes. So two labs have been doing a really fantastic job. The Schultz-Liefert's lab in Germany and Jeff Dangel's lab in North Carolina in making defined microbial communities, focusing initially just on bacteria and using these synthetic communities that they've determined that these microorganisms are usually present in the roots of a particular plant. From establishing a baseline, like a core community, you can go in and start to remove members, change the ratio of particular members, and look at what are the repercussions of this from how the community shifts, but also from the phenotype of the plant, from the appearance of the plant.
So tying the two together, the reductionist approach and the more ecological approach. We have another question from the audience.
It's actually from the Twitterverse.
Okay.
Somebody tweeted in a question, and I really apologize because I cannot pronounce his Twitter handle. But he asked, why do you pasteurize? It's not really a negative control, is it?
What? I'm sorry?
This is what he asked.
Uh-huh.
Why do you pasteurize? Is it a negative control?
So it's not really a negative control. So the normal plant is the plant that has microorganisms. The plant that, there's a session later in this conference called the Hologenomes and Holobionte Conference. Really the plant needs to be considered a holobionte, and all the microorganisms that live on the plant can almost be considered an organ of the plant. So the exotic thing that we're studying is the microbe-free plant. And we can infer what the function of the microbial community is by looking at the repercussions of removing it. And then selective introduction of subsets of microbes or different microbial communities, we can look at the differences. So it's not really our negative control, it's actually what we're probing, like it's the experimental condition is the microbe-free plants. So.
I think the analogy is people have learned a lot about the mammalian microbiome by having germ-free mice raised in very, very specific conditions that have allowed us to then learn about the more complex situation. And that's essentially what you've built. You've built the plant germ-free system that allows you to do these kinds of manipulations of one or more factors at a time.
Right. Yeah, and then I also think it's really important that there's more than one system like this, more than one microbe-free growth system to grow a plant. Because if we made all these broad conclusions about what the plant microbiome does in our system alone, it might not be reflective of what's happening in certain environments. So it's really important that other systems such as like a calcined clay microbe-free system or even hydroponic systems are also used.
Can we talk for a second about potential applications. I realize this may well be in the future quite a ways.
Right.
But once you understand more effectively how the beneficial, if you would, microbes in the microbiome prevent or help decrease sensitivity to pathogens, you could potentially artificially add those organisms to a whole field and change disease resistance in a way that does not require use of more nasty antibiotic-like compounds.
Absolutely. So, yeah, a good friend of mine, Carl Weinhold, is a coffee buyer in Colombia, and he goes to the growers and buys firsthand the beans. And he's told me he's seen firsthand the repercussions of this fungal pathogen called Roya, coffee leaf rust, that's really decimating crops of coffee. And this was a pathogen that completely ruined the coffee industry in Sri Lanka for some time back in the late 1800s. And now Guatemala, a couple years ago, declared a national state of emergency because of their coffee industry. And there's certain microorganisms, certain strains of one microbe called Bacillus subtilis that's able to fight off this coffee leaf rust fungus to some extent. But results are mixed. You could put it on one field and it's pretty effective for a while, maybe for a year or two, and then another field it just doesn't work. And then go to another field and it only works for a week or two, and then there's a rainstorm and it stops working or something. And we don't really understand exactly why the applications of these microorganisms are so, environment specific. But I think perhaps biocontrol, this is called biocontrol, could be more effective using communities of microorganisms. So understanding of how microbes interact in a community context could really lead to some more robust products for biocontrol, and also just for stimulating the immune system. Plants have an incredible immune system, and anytime you see a disease in a plant, it's because the pathogen successfully short-circuited the immune system or was able to suppress the immune system of the plant. So if we could use particular microorganisms to stimulate the immune system of the plant without a negative trade-off for growth or unwanted metabolites, it could be a really powerful tool, because rather than using nasty chemicals, you're telling the plant to use chemicals it can already make to defend against potential pathogens.
I think that the take-home points are virtually everything in life, plant, animal, you name it, has a lot of commonalities, and many of those commonalities are driven by the microbes that are such a core part of the world in which we live. And I think the other thing is how technologies, partly driven by sequencing technology, but the follow-up of that, is allowing us to really rethink the way that we can improve things like plant production, human health, and so on. So it really is a very exciting time to be doing microbiology.
Absolutely.
So, well, I want to thank our audience for joining in with us today. I want to thank all of you who are listening online for being a part of this and learning a little bit more about some of the really exciting and interesting science happening at the American Society for Microbiology 2015 meeting. Please join us again for another episode of ASM Live. I'm Stanley Malloy, and I look forward to an opportunity to talk with you again. Thanks.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.